Hello everyone, welcome to my YouTube channel shikshak. This video will be very helpful for all who are preparing for CSIR, UGC NET, Life Science, GATE, ARS NET examinations. Today we will discuss again a new molecular biotechnique, that is fluorescence recovery after photobleaching or FRAP. Now the question is that where we use this FRAP technique. We mainly use this technique for the diffusion analysis of lipids to study the diffusion of lipids and protein binding studies, and to understand the kinetics of diffusion through tissue or cells by the use of different fluorescent labels, GFV for the proteins and the P35 for the lipids, and also to study the membrane dynamics by forming artificial membrane in the labs. Now let us first recap fluid mosaic model. Because in this technique, diffusion coefficients of different molecules present inside membrane. In the membrane, the lipids are arranged as phospholipid bilayer. Phospholipids have hydrophilic head and hydrophobic tails. Hydrophilic heads point outwards while hydrophobic tails point inward of the membrane. And in the membrane, proteins are also present. These include peripheral proteins and integral membrane proteins, which consist of channel protein as well as carrier proteins. In this technique, we will study diffusion rates of lipids and proteins and other cytoskeleton elements present in the membrane, and will confirm the order of movement, whether lipids diffuse first or the protein diffuse first or the cytoskeleton elements diffuse first. This figure I have just added to revise is the concept of membrane fluidity. This figure shows that how lipids diffuse inside the membrane. In the figure A, flip-flop diffusion is taking place, in which phospholipid move from outer leaflet to the inner leaflet inside membrane. In figure B, this is an example of transverse diffusion. Here the diffusion occurs to the same position but with the help of flippase protein. In the figure C is an example of uncatalyzed lateral diffusion, in which lipid diffuse in the same leaflet from one position to the other position. Now you can easily understand that what is diffusion and what we are going to study in this lecture. So the answer is that we are going to study the diffusion rates of lipids as well as proteins and other elements, how they move inside the membrane when some conditions are applied, and that conditions we will discuss in the principle of FRAP technique. Up to this, I have given you the information regarding the basic idea of diffusion. Now we will discuss that what is FRAP. FRAP stands for fluorescence recovery after photobleaching. As the name indicates, first step is fluorescence and second step is photobleaching, and the third step is recovery. In the figure you can see there is a structure of plasma membrane. In the plasma membrane, lipids and the proteins are present. Lipids firstly tagged with the fluorescent probes. As the lipid contains phosphate groups, a radio-labeled probe P35 is used to tag lipids, and for proteins, fluorescent label GFP is used. So this was the first step to fluorescently label the lipids. And then the next step is photobleaching. This is done with the help of a laser beam. A laser beam is targeted on a small surface area on the membrane. After some time, the process of bleaching takes place. Bleaching means removal of fluorescent label at that particular area. So now you can observe the figure B. There is disappearance of tagged lipids in small area, but the rest of membrane lipids have fluorescent labels. As in this process, fluorescent label lipids move away and untagged lipid molecule take their place. And the third step is recovery of the molecule. When no laser beam is targeted on that area for some time, again diffusion takes place and the fluorescent level lipids regain their position. This appearance and disappearance of lipid molecules can be observed with the help of fluorescent microscope. Whenever there will be fluorescent levels all over the membrane, the fluorescent microscope will result in fluorescent reading as compared with bleached area where no fluorescent tags are available. There will be no fluorescence when observed under fluorescent microscope. In this figure, a graph was plotted. Fluorescence obtained versus time interval. Fluorescence obtained in the y-axis and time in the x-axis. These are again the previous experimental results but in graphical form. Now let us try to understand the graph. AB is the point at which lipids were leveled but not photobleached and full fluorescence was obtained. You can also correlate it with the membrane structure given in the right corner of the slide. And B and C is the point at which laser beam was targeted on small area as untagged lipid molecules appeared in this area. So the region between B and C represents decrease in the fluorescence as untagged lipid molecule diffuse in place of tagged molecules. After this, for some time, no laser beam was applied. And the region between C and D represents final step of the recovery. Again, tagged lipid molecules regain their original position. Due to that, fluorescence increases between C and D, and the region between D and E represents final recovery. Even after this step, some initially fluorescent labeled lipids were unable to regain their original position, means there was no 100% recovery. If we consider this recovery as 80%, there must be 20% of fraction of fluorescent level lipid molecules that was unable to regain their original position. This fraction is indicated by the immobile fraction in the figure, and the fraction that have recovered is known as mobile fraction. You must keep this information in your mind for solving the questions. In the figure, I have represented the diffusion coefficient for the lipids. Keep in mind, greater is the diffusion coefficient, faster is the recovery of the molecule. In this figure, I have added important point to remember that lipid diffuse first, then proteins, then cytoskeleton, and then cells that are not bonded to the membrane. As it is clear from the figure that diffusion coefficient for the lipids is greater than that of proteins and then that of cytoskeleton elements and that of cell which are not bonded to the membranes. Hence, the cells which are not bound to the membrane will take the more time to diffuse. Now, let us try to solve some question based on this information. In the question, it is given that you have fluorescently tagged a protein X using antibodies and perform the FRAP experiment. Graph is just the result of FRAP technique. Protein X is represented as red in color. When we have compared the diffusion coefficient of the protein X with that of control, we have observed that protein X is having the low diffusion coefficient as compared with control, and control is having more diffusion coefficient. So the protein X will move slower. Now let us solve the next question which appeared in the CSIR UGC net 2016 December. The question is fluorescence recovery after photobleaching. FRAP is a method to estimate the diffusion of molecules in a membrane. Fluorescently labeled molecules such as were used. A receptor tagged with green fluorescent protein was used. Number 2, a receptor labeled with GFP which interacts with cytoskeleton was used. Number 3, a labeled lipid was used. And number 4, a labeled protein that binds to the membrane surface was used. After that, the researcher have plotted a graph from the FRAP technique results data. In the figure, they have given the result of that data in the graphical form, and they have represented a point B, A, C, and D in the graphs. They have asked us to correctly match these points with that of correct tagged molecule. as it is clear from the data that they have used lipid tags, protein tags, and cytoskeleton elements tags, and I have already told you the order of movement of these. For just to remind you all, the order was lipid, then protein, then cytoskeleton element, then not membrane bound organelles. Hence, in the figure, point B will represent the lipid, and point A will represent the protein, and point D will represent the cytoskeleton bound, and D represents the elements which are not bound to the membrane. Hence, we will pair B with the third statement, which represents lipid diffusion. As A point represents protein, we will pair it with the fourth statement, and the fourth statement is in which a labeled protein that binds to the membrane surface was used. Hence, the right answer is answer B. Now, let us solve another question which appeared in the CSIR UGC net December 2019. Question is to investigate the dynamic nature of two unrelated centrosome localized GFP tagged proteins GFP-A, GFP-B. A team of scientists conducted fluorescence recovery after bleaching experiment. That is, they have conducted FRAP experiment and they have given the FRAP graph results. Now, based on this graph, we have to find the right statement. Let us take a look on A statement. It is given that GFB shows faster exchange rate than GFB-A. In the statement B, GFB-A shows faster exchange rate than GFB-B. In the statement C, GFB-A has more immobile fraction than GFB-B protein. In the statement D, GFB-B has more immobile fraction than GFB-A, and they have asked that which of these above mentioned statements are correct. For your information, they have used two proteins to study the diffusion rates. Proteins are GFB-A and GFB-PA. From the figure, it is clear that GFB-B have the more diffusion coefficient. Hence, it will show the faster exchange rates than that of GFB-A. Hence, it is clear that statement A is right. And the second thing they have asked us to observe is the immobile fraction. And from the figure, it is clear that protein GFB-B is having the low immobile fraction than that of GFB-A protein. GFB-A protein is having the more immobile fraction. Hence, the statement C is also right. Hence, the right answer is answer number 1. Thanks for watching this video. And don't forget to subscribe to my YouTube channel BioShikshak, and do hit the like, share and subscribe button.